All right, hello guys. This video is for will be our mitosis notes. So if you look at our cell notes packet, you should already have this portion completed, which is just on the big picture on the cell cycle mostly. And then you should have the vocab terms completed by now. And then if you scroll down to the beginning of page 3, you'll see there, this is where we are. So we're gonna only focus on mitosis. So make sure that you're filling in these notes as you're listening to this lecture. So I know that a lot of you have learned about mitosis in middle school. And I think it's a little bit deceiving because at that point you kind of picture that cells are always doing this and that this is what they do. But if you'll recall from the last lecture, this is really just a tiny portion of what cells are actually spending their life doing, right? So most of the time the cells are in interphase. So there's a lot of little details, a lot of steps along the way within this process of mitosis, but I swear I really think that your notes, the way your notes are laid out, I think it makes it really manageable. And really you can see that some of the steps, there's only really a one or a few main components that you need to memorize. So cell division is the process that we use to identify the actual process of one cell dividing into two. Okay, so actually cells dividing and making new cells. Each body cell that results from cell division must have an exact copy of the DNA in the original cell. And what I mean by that is, as I'm sitting here today making skin cells, my skin cells want to make identical skin cells, and they all have to have a full set of DNA. Okay, you can imagine that there'd be a major problem if skin cells, as is, just divided into two, and then each of the daughter cells had half of the DNA. That wouldn't make any sense. So instead, what has to happen is sometime in the process, remember during the S phase of interphase, the DNA actually gets replicated. And at that point in time, our cells, remember during interphase, our cells are just acting like normal cells. So skin cells are just acting like skin cells. During the S phase, the DNA is replicated. It goes through the G1 phase, and then at some point in time, it's going to be triggered to divide the cell into two. And because the DNA has already been replicated, each cell is going to have a complete set of DNA. So here is an image that shows the whole cell cycle. So you can see here on the inside, you can see the G1, the S, and the G2 phase, and we call those interphase, right? And that's when cells are just acting like normal cells. Along the way, they're doing some extra stuff. So remember G1, if you imagine this is a brand new cell coming out of mitosis, a brand new little daughter cell, pretty tiny. So it's going to bulk up. So G1 truly means like growth. They're going not only like in girth, like literally getting bigger, but also reproducing organelles, anything it has to do in there to just kind of grow and be more successful. And then it's going to be triggered at some point to enter the S phase. And remember, we call this like the synthesis phase. So DNA is replicated. So we literally go from one complete set of ribosome DNA and double it. So now we have two complete sets of ribosome DNA. At this point, the DNA is still stuck in the nucleus. Cell is still really functioning like a normal cell. Eventually, it'll move on to the G phase. And remember, G just means growth. Again, another phase of growth as it gets ready to prepare to potentially divide into two. After all of this, for whatever reason, and maybe just general growth, and maybe because I got a finger cut at lunch, but for whatever reason, that cell now is going to be triggered to enter mitosis. And mitosis is really just the explanation of the phases of how one cell divides into two. Okay, you can imagine that there's a lot that has to go perfectly for the cell to survive. and perfectly divide into 2. So now today, from here on out, we're really going to just focus on those mitotic phases. Here's the vocab that you've already mastered by now. So let's focus on mitosis. So the first phase of mitosis is prophase. Prophase. What we have happening in prophase is the centrioles. Remember, the centrioles are the little pill-like looking structures. In the image here on the bottom right, the centrioles are represented by these little pink structures. So here's a set of centrioles, and here's a set of centrioles on the other side. And at this point in time, they're going to separate to opposite ends, start moving to opposite sides of the cell. From them, the spindle fibers are going to begin to form. So you can kind of see like these little pink lines. Those are showing representing the formation of the spindle fibers. The chromatin. So just let me get my handy dandy. Remember I've said that most of the time our DNA actually looks like this, which is chromatin. All during interphase, when our cells are functioning like normal, our DNA looks like this, a big old blob. Here at the end of prophase, you guys, is when the chromatin is going to start to condense, start to form chromosomes. So like if we're looking at real cells in a microscope, sometimes we'll call we'll call a cell prophase and you can't see any chromosomes. Sometimes it'll be just like see the beginning of chromosomes beginning to form. And remember from last lecture, I said that I call like real chromosomes and real cells, they look like little wormy looking, dark wormy looking things. And we'll see some more pictures of that. And then the last thing that happens towards the end of prophase is the nuclear envelope begins to break down. Up until this point, the DNA is stuck in the nucleus and it stays safe there. All during interphase, our DNA is stuck in the nucleus. What's going to have to happen in order for the cell to divide is the nuclear envelope totally dissolves, and that's what is going to allow the DNA I call it, they have free range in that cell, and we'll see why in just a second. So if you look here, this kind of dark purpley line normally would be solid, and that would be representing the nuclear envelope. You can see now that it's a dotted line, and it's a dotted line because it's representing the fact that it's kind of starting to get broken down and dissolving away. Okay, oops, sorry. So again, in prophase, the beginning of mitosis, the centrioles are moving to opposite ends. The centrioles are beginning to form spindle fibers. The nuclear envelope is beginning to dissolve. Oh, and the chromatin is condensing into organized chromosomes. Here is a real-life picture of a cell. And again, I can tell this is prophase because I can see the chromosomes, right? Do you see the little black wormy looking things? But they're still condensed in the nucleus, right? So almost all the time, if you see a big old regular cell and in the nucleus, there's very obviously still a nucleus, but you see the chromosomes there, that tells me it's probably late prophase, where the DNA is beginning to get organized in the chromosomes, but they're not spread across the cell quite yet, which we'll see next phase. Okay, so that's all prophase. Metaphase, really just pretty much just one simple thing that I want you to remember with metaphase is that the chromosomes meet in the middle. Okay, so metaphase, they meet in the middle. Whole bunch of M words. The chromosomes are moved there. Remember, they're attached to the spindle fibers, and the spindle fibers kind of shimmy and shake and organize the chromosomes. If you look at this diagram, oh my gosh. Oh my gosh, I'm recording a lesson right now. Oh my gosh. A really adorable dog just walked in. How do I pause this, Emily?
Oh no. Okay, we'll just leave. I just wanted you to meet Archie. Just keep talking about spindle fiber.
Oh no, just a second. All right, sorry about that. A student I don't even know just brought in the most precious dog. Oh boy. So they saw me just melt on the floor and let that dog just lick all over. Real highlight. Okay, so anyways, with metaphase, the chromosomes meet in the middle. And remember that the chromosomes are moving throughout the cell, all thanks to the spindle fibers. So in this picture on the bottom right, you can see a cell here. You can see the centrioles. Coming from the centrioles are those spindle fibers. And then the spindle fibers have attached and lined up like almost, they really do almost perfectly line up those chromosomes in the middle. On the picture on the left here, you can see these are plant cells. So like the yellowy brown color is showing you the cell wall. So the cells are rectangular shaped here, and I don't know what I was going to say. But this cell right here, you can see it, I'm trying to make it really obvious, is definitely in metaphase. So this is, I mean, normally we'd be doing labs with microscopes and you'd be going over there with plant cells. You'd make your own little slide with plant cells. And you guys could truly identify metaphase. So you guys can see that these black wormy looking things are the chromosomes and that they're truly just lined up in the middle. I'm going to guess, just so we're here, if you guys look at the cell to the left of it and to the right of it, if you check out that nucleus, do you see how the nucleus itself is just shaded black? You cannot identify black worms, right? That means that I'm going to say almost positively that those cells are in interphase, because the DNA just looks like this, right? So there's just like a hue, a shaded in black of the DNA that's totally disorganized. So metaphase, they meet in the middle. In anaphase is another pretty easy one. So in anaphase, they move away from each other. So the sister chromatids are now separated, okay? So one is going to be pulled to the left and one is going to be pulled to the right. So in anaphase, they move away from each other. In the diagram on the bottom right here, you can see that one of the yellow chromosomes is being pulled towards this end of the cell, and the other chromatid is being pulled towards this end of the cell. And it's pulling one complete set. Can you see that? So there's one full set of this cell's DNA is going to this end, and then one full set of DNA is going to this end. And remember, that's ideal because ultimately we want two chunks of identical DNA, right? We want a full set of DNA here and a full set of DNA here. There are errors. It's not super uncommon for an extra chromosome to get pulled and then this one to be short, right? Most of the time, our bodies are really good at actually identifying that error, and our cells, our body literally like breaks down the cell and kills it. We'll talk about some genetic issues when cells have, or individuals have an extra chromosome or missing a chromosome, and we'll talk about that. And then in the bottom left is a photograph again of plant cell under a microscope. And here you can see, again, the rectangular cell wall. And you can see that the black wormy chromosomes are physically being pulled away from each other. So that's going to be anaphase. There's more pictures. Another really good one. The photograph here shows the chromosomes being pulled away from each other into two groups. And then some people call this the last phase. The fourth phase is called telophase. And telophase is really, it is the last phase where one cell has two nuclei, two identical nuclei. So let's see what happens. In telophase, the chromosomes gather at opposite ends of the cell and slowly decondense back into chromatin. So towards the end of telophase, we will no longer be able to see the chromosomes, and instead we'll be back to seeing chromatin. Two nuclear envelopes are going to slowly start forming around each clump of DNA. So here's one set of chromosomes, and you can see the dotted line again. So the nuclear envelope is now reforming. And notice that it's going to do the same thing around the two sets of chromosomes. The spindle fibers break down. The spindle fibers, those pink lines that were moving across the cell, they've done their job. Nothing else needs moved or shimmied around. So the spindle fibers get broken down. Notice that in telophase, these two nuclei are still within one cell membrane. So this is still what we consider one cell. The last phase is cytokinesis. And the cytokinesis is the quickest of them all and is really just the cell pinching in two. So this is the final stage of mitosis. Sometimes kind of people sometimes pair this with telophase. So they'll say like, and then telophase and cytokinesis, because honestly, cytokinesis just happens so fast. And really the only thing that happens is it's the cell officially pinches in two. That's it. Each daughter cell has its own nucleus with an identical set of chromosomes. So they should be identical as each other, but also identical from the original cell that it came from. And remember, this is not making babies. This is cells everywhere else in our body. So liver cells right now in your body are making new cells just for regular maintenance. So you want a liver cell, a complete liver cell, to end up with two identical complete liver cells. So cytokinesis again is like literally the pinching of the cytoplasm, okay? I love this picture on the top right. It's a real photograph of cells. You can see they're just hardly being held together here in the pinching of the cells. Here is a microscope image of cytokinesis and in an animal cell. And we can tell it's an animal cell because it's round, and you can tell already that the cell membrane is pretty flexible and squishy here. And in fact, that pinched area that the blue arrow is pointing to, that's what we call cleavage. And so you can guess why the term cleavage gets thrown around society. So basically what happens is the cleavage begins to just become more and more and more, pinched, and it just keeps continuing to grow until the cell divides into two. Now here's the trick. Animal cells can do that because our cell membranes are so flexible. What happens with plant cells is totally different. So plant cells, because they have the rigid cell wall made of cellulose, their cells cannot and don't pinch in two. What happens instead is the plant cells actually grow what's called a little cell plate. I love this picture on the top right. I should have made it bigger. So you can see the original cell wall is this like dark red rectangular shape. This cell has undergone mitosis. So you can see the two complete nuclei. And to me, it looks like the chromosomes are just starting to kind of decondense. You can kind of see some wormy stuff, but maybe just chromatin in there. And then what's happening in the middle, you can see it perfectly right here. What's happening in the middle is literally a cell wall just begins to grow in the middle, and it begins to grow further and further and further and further all the way out to the cell wall. Eventually, when the cell plate reaches both cell walls, cytokinesis has been completed. So even like this purple picture here in the middle, you can see the new cell wall right here. Obviously that new cell wall is going to be pretty thin initially, so it'll take a tiny little bit of time to actually develop into like the thickness of a regular cell wall. There's mitosis on donuts. Okay, so we have gone over a lot of those structures together. You remember the cleavage furrow, you remember cytokinesis in animal cells here versus plant cells. Remember that plant cells form a cell plate instead of the cleavage furrow. Oh, it does move. So here we go. This is mitosis in an animal cell. I can tell because of the cleavage furrow. So here the chromatin is condensing to chromosomes, the nuclear envelope, disappearing. Chromosomes are meeting in the middle. They're pulled away from each other. They're gathering in two groups and it breaks apart. So here we have prophase. They're going to meet in the middle in metaphase, pull in anaphase, pull away from each other. Telophase, two nuclei in one cell, and cytokinesis when it breaks into two. Here are some good pictures of real cells in mitosis in plants. So you can see in this plant cell, here's the nucleus. We're going to call this like early prophase. You can still very much see that the DNA is still in chromatin, just starting to condense. In late phase prophase, we can start to see the black army chromosomes. Eventually, the nuclear envelope is totally gone, and here in metaphase, we can see that the chromosomes are meeting in the middle. In anaphase, those chromosomes are pulled away from each other. In telophase, we have two noticeable clumps of DNA. And in this plant cell, right in the middle is a beautiful cell plate. Remember that cell plate's just going to continue to grow and develop until it reaches both ends and functions as its own cell wall. So here's prophase. Metaphase, they meet in the middle. Anaphase, they pull away from each other, and then telophase, anaphase, telophase and cytokinesis are tricky in this one. It goes pretty quick. Okay. All right, you guys, that is the end of your mitosis notes.